So which cells? There's a lot of cells in a microenvironment. A tumor is not just made of tumor cells, it's made by a lot of other cells. And in fact, almost all the immune system is implicated with these proteins. So for the first one, COCA1, we see it in parts of the immune system we call the innate immune system, and we can see them, and since actually my clinical background is an immunologist, I love to be working on this one, but we can see it in, we can see it in the eosinophils and the big granules of eosinophils, mast cells. These are allergic cells, but we have them in the gut too. We can see them in neutrophils, macrophages, and dendritic cells. So an arm of the immune system, the myeloid arm, actually expresses COCA1. And interestingly, I mentioned that we can see it in the granules of these cells. We actually can see, and it's just hard to see with this level of, without a microscope, but we can actually see that they're in the granules of cells, but we can also even see them that they're actually in granules which are outside of cells. So we used to learn in microbiology that these cells would just liberate their proteins, allergic proteins and so on, right into the media. But in fact, they can actually secrete little granules, and we can see the little granules which are outside of cells going through between tumor cells and so on. So we have extracellular granules containing COCA1. And for COCA2, we actually see them in all the cells. They are not in the granules. Now they're in the cytoplasms of cells, but we can see them in immune cells. These are lymphocytes, epithelial cells, endothelial cells making a vessel, and so on. Again, there's many cell types, but as we had this COCA2 marker, we seemed to believe that we could see clusters of cells which look like lymphocytes. So rather than look at COCA1 and 2, we decided to look at lymphocytes. And we can see this is a lymphocytic marker, an immune cell, and you can see there's an awful lot more, keep on pushing the wrong button here. There's an awful lot more red in the top panel than the bottom panel. Again, so we had with COCA1 overexpression of one protein in the myeloid cells, and with COCA2, and now with this lymphocyte, we can see there's a higher lymphocytic infiltration. That's interesting because pathologists for 20 years have actually described that having more lymphocytes in colon cancer tissues is actually a sign of better prognosis. And this is to say when you go and do large numbers, it's easier to express. So this is called TMA analysis, where we can compare the expression of the different proteins compared to the 3 genotypes. And this is AA, which is protection, and CC, which is risk, and these are heterozygous. So you could basically see that in larger tumors, so it's reproduced in large numbers of samples for COCA1. For COCA2, both in the microenvironment, the lamina propria, and the epithelial cells, we see this association. And then in this is a lymphocyte marker, again, to show there's more lymphocytes if you have correlate with protection. This project is 2 years old, and I still don't know what these proteins do, I have to be honest, but it certainly gives us an idea how we can take genetic information from these studies and start exploring new areas of biology, in this case, innate immunity. So just to summarize, we started with a genetic association at a region, which we were able to compare between case and controls. The variant was found, and then the genetic variant led us to the nearby genes, and we could see the nearby genes overexpressed correlation with protection. And then we were able to see that the proteins are in the granules of cells, in immune cells. And again, 5 minutes? Okay. I'm going to go to a clinical trial. I'm going to study. So right now I was talking about genetic variation. I was giving too much explanation. Genetic variation in the tumor cells has a lot of mutations, as Eric showed before. So this is an abnormal karyotype. This is a map of the large projects which OICR is coordinating in terms of resequencing cancer genomes. So we have about, actually will be 52 by next week, of large projects to sequencing 500 tumors across the world. The informatics for the project is coordinated at OICR so that we actually host a little database at each of the sites participating in the project so that we can actually put the information available, link it up, and make it available to cancer research labs. And on the website, we have information for 7,000 tumors, and this information is growing very quickly. We had 1 cancer genome in 2007. Now we have about 7,000, so we should be over 10,000 very soon. And there are lots of mutation genes. So what I thought I would basically try in my last 3 minutes, say there's a category of mutations which we like to call actionable, which are linked to drugs in certain types of cancers. And slowly we understand that even BRAF mutation is not just a melanoma. We see at some percentage in every cancer. Herceptin is seen in many types of cancers. And EGFR for lung cancer is seen in many types of cancers. And it became our idea that it would be important to test, make a diagnosis not on which tissue the cancer comes from, but what are the drivers, the cancer mutations which are actionable in any cancer. And I'm going to, so this skip of drugs, basically we've gone to the concept of doing a bit like infectious disease. I see Michel Bergeron here. We don't decide, today what we're doing a single test at a time in genetics, and if you were to do it in microbiology, you'd say, I think it's a staph. You wait for the results, and if it's negative, you go for a strep, and so on. No, you look for the whole set of bacteria that might give fever, and here you look at the whole set of actionable mutations before you make a selection. So it's very different the way pathology labs, which are still doing single tests. And so we've actually embarked on a study. Andrew Brown's in the room as a postdoc who's been doing this. But first we started taking the tumors of patients with metastatic disease beyond standard of care, had received over 3 drugs on average. And so there's nothing left. And we started a project, involves 5 sites in the province from Thunder Bay, London, Kingston, Ottawa, and Toronto, where patients which want to get their tumor sequenced, be part of one of those trials, get it analyzed, and then we look for those mutations for which the drug exists. And the fact is, we're at this point because we've used different technologies, which I'm not going to get into, but these red dots here means the number of patients which have been impacted. So these are patients for which a mutation was known to exist in that type of cancer, but not only had we detected, we were able to predict that they might respond to a drug. And so the red dot is about 20 patients. About 30% of patients have an actionable mutation. The second part, I can only talk about the ones for which have more than 1 year of data. It's the feasibility study, which was just published. And I'm going to skip the technology to finish with one slide. Yeah, this one here. Sorry. Is that patients which are beyond standard of care, when you sequence their tumors, you do identify mutations, PIK3CA mutation in colon cancer. There's a drug that exists for that in clinical trial setting. AKT mutation, this is the first time we saw this mutation in breast cancer. RET mutation, well, thyroid cancer might have been known. EGFR mutation is a novel mutation. And these are patients for which we are able, because of our large clinical trial network, to try the drug, and 4 of 6 patients had a clinical response. So this is just the start of showing how genomic information applied in the clinical setting, in a clinical trial setting, is able to predict responsiveness to potential drugs, give them the drug, and we're starting to see clinical responses which had not been anticipated, were not part of clinical studies elsewhere in the world. So there's a lot of potential. Now, I know I'm running out of time, but I think that the key thing is not only do we need to do larger numbers of studies like this in Canada, this AKT mutation which we saw, we'll maybe never see it again in Toronto. The next time it may pop up might be in France or Heidelberg or at the Dana-Farber. It's important that, as we did for Genome Project, for HapMap and ICGC, that the big clinical trials groups doing these types of studies are able to share information about mutation, treatments given, interventions, so we can start up building a body of knowledge which is not just based on anecdotes, but larger numbers. And again, this is what's going to help us to be able to optimize the treatments of patients in the future. Thank you.
Do we have some questions for Dr. Hudson? Please.
I'm just curious about the availability of those personalized clinical interventions.
Okay, so what we work in, so I notice now I have 5 minutes to answer questions. So keep the questions coming. It's a really good question. Right now, most of those drugs that we're testing are available only through clinical trial setting. So these are novel drugs which haven't yet been approved by FDA and so on. Some of them, of course, have been, like I have the list, but many are not. So what we do, because we're a Translation Research Institute, we do a lot of clinical trials and so on. So we have about 300 trials open in the province. We actually have set up a clinical trials unit at every one of the cancer centers in the province. And all the university medical schools, they're participating in this study. So generally, physicians in the province are now very attuned to this project. They're on the call at Wednesday noon every week listening to what's happening. So we have groups in Thunder Bay, as I mentioned, and so on. And when they think they have a patient which is beyond standard of care, they've done everything and are now eligible for clinical trials, they contact the clinical study and they participate. In the future, though, in 10 years or 15 years, I suspect there'll be a lot more of this information will be available to doctors, and these are a list of drugs that exist that are FDA approved or Health Canada approved, and this is a list of tests that can be done. But it will be, again, a learning to match, optimize the treatment based on the tumor, on the molecular profiles of the tumors. So that's what I think the future is. This is how we can see what the future is going to look like. Today, you have to be part of a clinical trial setting.
Thank you.
Thank you. I always enjoy the presentation, Tom. It's excellent. I have a question regarding the first part of your talk. Maybe I'm not in the field. So the protein you identified, COCA1, COCA2, is it known already, or is it known proteins already in the literature?
No. Because I've been part of the genome project since '91, since the beginning. And we always talk about the genome at different levels, but the fact is, even if with the whole sequence of information, I'm as a genome scientist, so I look at it, most of the information that's encoded by the genome is given, we still don't know what it is. We know that this, because of the ENCODE project, that this region might be regulatory, might be more active in lung than in liver, but we really don't even know in some very cases where it's involved in development or only when there's an infection and so on. So we still know very little. So for those cases, there are a lot of proteins in the genome, we can predict them because they're spliced, because there's an opening reading frame, so the genetic code predicts that they would make a protein, but we don't even know anything about them. So this is a case of not having known. Had I found that those genes were only expressed in the big toe, but not in the colon, I probably wouldn't be working on them. But that's the level of information. We don't even know that as we're getting in, and there's a whole lot of the genome biology which is still not explored.
But you have, I may have missed it, you have done tissue microarray on these things.
There's an echo, I can't hear you well.
Tissue microarray, TMA analysis on a colon cancer? Did
TMA?
I miss that? Yeah.
Yeah, this was the plots I showed were TMA analysis, yes.
Do you see what percentage of the TMA that you see your marker? Is it possible to see that?
Well, the red, you can see there's many ways of measuring positivity, but it's about 1 or 2% going to 5 to 10%. And that's the kind of the scale we've seen. So there's a lot of the cells around the tumors, which are, again, immune cells, something which we've known for a long time.
Okay, thanks very much.
Thank you. Please.
Yes. Just a question about the large amount of sequencing that's come out of the primary tumors. I don't know if you've looked at it to see whether there are additional DNA markers which might be useful in early detection, which is, but I'm not sure everybody appreciates that colon cancer is one of the prime candidates for cures associated with early detection.
Okay, so in terms of the genetic markers that are inherited, the germline markers, the ones that we know which are associated, there's two subgroups. So the ones we've known for about 15 years where DNA repair genes, which are mutated, give familial forms like a dominant disease. And these are available for testing. They're rare, but they do account for familial forms. In terms of sporadic forms, the most common types of colon cancer, I think the only ones which are published right now are those 10 plus the next 10 that came in the last year. So we have a list of what's known today, but as we're doing larger and larger studies, we're finding more. So we haven't finished exploring what that space is. For prostate cancer and breast cancer, which are more advanced in terms of large studies, there's about 100 risk variants across the genome, which are well replicated in thousands of individuals, which are risk factors. So your risk is not a single gene like the familial forms. It's actually a dosage of the more alleles you inherit with risk. Again, the higher the people with that number of alleles have over others. So we can see population risk increases with numbers, but the risk is confirmed by hundreds of spots. In terms of the tumor mutations, again, we can see generally when we sequence a tumor genome, about 10,000 somatic mutations. I've seen some with 200,000. We keep on talking about KRAS and so on, but there's a lot more mutations. In genes, protein coding, there's about 100 in every patient. there's only about a dozen, which we see in more than 5% of patients, like TP53 and KRAS and so on. Most of them are very rare, and to actually be able to detect whether it's a driver mutation or just a random DNA error, it takes these large numbers that we're trying to do with ICGC. And I suspect that at the end of the game, we will have some strong, just like for genetic disease, we have some strong drivers, give Mendelian forms, and we have all these small risk variants. I think then in a tumor cell, there'll be hundreds of drivers, a few strong ones like KRAS and BCR-ABL and so on, but also hundreds of them which give a small growth advantage, evading immune system, better metabolism and so on. And that strength of the cancer cell is unique, not only because of one big mutation, but a whole lot of other biology that's been affected by some of the variants in that tumor.
Good. We have time for one more short
Hi, Tom.
question.
That was
and
an
answer.
excellent talk. So I have a question towards, I'm very impressed about how you've taken this into the clinic already. I think it's early days, and it's impressive that you're impacting patients. So I was wondering, where will you be in 2 years? So, and it'd be great if you could speak to the barriers. I mean, I heard sort of between the lines, there's cost and there's also speed. Are there any other barriers that are crucial?
Well, I think one of them is a cultural barrier, and one of them is cost. So there's more than one, but the cultural barrier is basically causes fragmentation. Everybody wanted to do the same thing on their own, and using a slightly different protocol, different things, different technology, and so on. Figuring out they're going to identify, understand cancer on their own. In terms of cancer genetics, it's not going to happen that way. There's not enough cases in Ontario of cancer to understand all cancer mutations and the right drugs and so on. So I believe that as we've done for ICGC and HapMap and other projects, we need large-scale collaboration, which we're able to still generate the data and have some innovation in our design. But the key information that matters in terms of mutation, validating mutations, drug therapy, and response can actually be shared, so we start building up some body of knowledge around those mutations, which are larger. And from where you see signals, then you can define a drug company may want to develop a really a full clinical trial with controls and cases, randomized and so on. But getting access to the signal and so on is important, and the large numbers have to be big, and I think it's through collaboration. The second one is cost, and I actually don't have the answer because we know these cancer drugs are rather expensive. There's different approaches to subsidizing drugs and having access to it. Right now, it's the fact that we're part of a clinical trial setting that makes us having access to the drugs, and the drug companies provide us the drugs. And so there's a certain benefit. We can actually show that the drugs are actually useful in other indications than what they thought of, and we have access to the drug for free, at least in the context of the trial. So it's kind of as a academic/company partnership, but we're not working with a single company. We're working with all of them as long as they're interested in providing their drugs. So it's kind of that's the space we're living in today. In the future, I mean, we're trying to solve things. Just so clinical trialists can accelerate this. We're trying to build up a pharmacy, and there's a lot of discussions between NIH and Cancer UK and other Canadian groups in having a pharmacy that the clinical trials groups, which were able to do all of these trials, have access to the drugs quickly. And I imagine that we will have a pharmacy for these types of studies available to groups in the few years.
Thanks.
Thank you very much, Dr. Hudson.